request presenters SITI, FATIMAH, and Nurellya and Hann Yih to raise your hand so we can promote you to the panel. Who is J2J? Can you rename properly before we promote you? Can you rename with your actual name? We're still looking for Nurellya and Hann Yih. It is easy for us to identify if you rename your Zoom name properly. It's kind of delaying the start of the session. If you quickly identify yourself and rename your Zoom ID name. We have 4 presenters already in. We're still missing 2 of them. Okay, let's promote J2J. And Ibrahim. Who is Ibrahim Abuga? Are you a presenter for today? I don't think so. Yeah, these are the presenter list. Alina, can you go to the next slide? Just for them to show, and then we come back to the judges slide later on. So we have 4 presenter here. 5, is it? 1, 2, 3, 4, 5. I think we're still missing 1. Nurellya, we are missing. Is Nurellya here? Ilya is Nurellya. Okay, can you raise your hand? Okay, we can start the session. All right, very good morning to everyone. Assalamu alaikum and welcome to this oral presentation finalist round 1. I would like to again welcome everyone for this conference. I'll be chairing this session, and we have 2 honorary judges, Associate Professor Dr. Elizabeth Davis from Monash University, Australia, and we have Prof. Dr. Ong Ching In from International Medical University, Malaysia. We will start this session, just some ground rules. Can we go to the next slide? We have 6 presenters. As you can see, your oral presentation code as well as your name and affiliation, and we'll follow the sequence as given on the website. And for each pre-recorded video, there are 7 minutes followed by 3 minutes of Q&A. And please keep your answer very specific to the questions, so we end up in time. All the panels, especially those who are presenter, are requested to keep their mic and video off unless they are answering the question only when we ask. So meanwhile, you please keep your mic and video off. Okay, with that, I think we'll move to the first presenter of the day, Ng Pei Yuen, Dr. Ng Pei Yuen from UKM.
Hi everyone, thank you for listening to my presentation. I'm Pei Yuen from the Faculty of Pharmacy, UKM. I would like to present one of the work in my lab, which is mainly conducted by my PhD student Amnani. So this is part of her PhD work. The overall concept of the project is on drug repurposing in oral squamous cell carcinoma. And in our study, the most promising hit that we found is galdanamycin with the structure as shown over here. And we also found that it could act synergistically together with cisplatin. So as we all know, cancer is a population of heterogeneous cells, whereby there could be a minor population known as cancer stem cells. These cells may not be responsive to standard chemotherapy, such as cisplatin, which is commonly used in the treatment of oral squamous cell carcinoma. So the survived cancer stem cells could then undergo self-renewal, which then allows it to grow and develop into a new tumor, which then causes cancer relapse in the patients. So, and we also know that there's not many new drugs available to treat oral cancer. Therefore, testing on the existing approved drugs could provide a faster route in implementing the effective candidates into the clinical trials. Therefore, in this study, we aim to rapidly identify the candidate compounds that can inhibit the cell viability of tumor spheres derived from OSCC through a high throughput drug screening process. To make the cancer model that we test more relevant, so we have grown cancer stem cell-like cells, which are also known as tumor spheres, from different patient-derived cell lines. But in the end, only the SAS cells could grow consistently into sufficient amount under the low attachment condition in also serum-free medium supplemented with additives as shown over here. So the sufficient spherical tumors grown allow further subculturing into 96 well plates, which is then could be performed on the drug screening phases as shown later on in primary, secondary, and tertiary screening, and it's being analyzed and tested using the MTS assay, right? So before we actually conduct the drug screening process, we actually studied a little bit on what are we actually testing on. So we did some. So this is how they look like under the microscope. So this is a diameter. So it's a little bit huge in micrometer size. So we also did some transcriptomic analysis, which is then performed using the Clarum S assay, which then revealed that there's various genes involved in the metabolism of fat and glucose, and some of the signal transduction pathways members are significantly being upregulated. So this data is then validated by performing qPCR on the highest upregulated one, which is SPT-SSB, which is the full name known as serine palmitoyl transferase mod subunit B, and also on the most down-regulated gene CCL2, which is also known as the CC motif chemokine ligand 2. So you can see that the data actually tallies. And we also have a look at the stem cell markers, which is the SOX2 and OCT4. So yes, the gene expression has been increased as shown in the qPCR model, and also at the protein level as shown through the Western blot. So we therefore conclude that the model is sufficiently cancer-ish like. Right, so we then move on with the primary drug screening. We did it on actually 1,463 drugs which are FDA approved or those who are already in the clinical trials, whereby in this phase, it's only one well being tested for each drug at 10 micromolar for 72 hours using MTS assay. After correcting their interplate variability, B-score normalization is used to filter out the hits. So the hits are, are they actually defined by B values lower than 3 MAD, which is whereby MAD actually refers to the median absolute deviation. So from the 174 candidates, we further analyzed using the stitch method and to recategorize them based on the mechanism of action and their currently available evidence, which have been published in cancer studies. So we then further select 8 drugs for the secondary screening. So here we just show a part of our data that we do found that galdanamycin has the lowest IC50, which is at around 28 nanomolar, and we further test them at different concentrations with cisplatin, and we've got a combination index analysis of 0.66, which suggests that it's a synergistic effect together with cisplatin. So in relation to the mechanism of action, galdanamycin actually binds to and inhibits the cytosolic chaperone protein functions of the heat shock protein 90, so which is HSP90. So it's originally discovered as an antibiotic. So it can actually promote proteasomal degradation of oncogenic signaling proteins, which may be overexpressed or overactive in the tumor cells. However, galdanamycin is only in the clinical trial phase, so as further evaluation can be done with galdanamycin and also with the newer members of its group, such as gantenerumab, and we can also test the interaction, the mode of interaction with cisplatin, and also further test its in vivo efficacy as well. So that's all for my presentation, and I will also use this opportunity to thank the funding body, which is mainly by my university, UKM, and also thanks a lot to my collaborators within UKM and also outside UKM in IMU as well. Thank you for your attention.
So can I invite Prof. Leaves?
Hi, that was a lovely talk. Can you tell me a little bit more about galdanamycin and where it comes from? Is it a synthetic compound or is it a natural source? Sorry, you may have said this.
Hello. Hi, Elizabeth. Thanks for the question. Can't remember directly. It's a very complex structure, so I think it's from the natural compound base. Yeah. But it's also one of the earlier, as mentioned, it's one of the HSP inhibitor, heat shock protein inhibitor. So it's actually one of the earlier generations. So there are the newer ones which are more synthetic based, which we have yet to test on to validate the more, because the newer compounds it probably has a higher potency, isn't it? So that we could probably study further in the future as well.
Okay. Thank you.
Okay, all right, let's move to the second presentation, which is by Ms. Goh You from International Medical University.
A very good day to everyone. I'm Goh You, a fourth-year medical student from International Medical University. Today, it's my great pleasure to share with you our study on the effect of tocotrienol-rich fraction on retinoscleral apoptosis in rats with streptozotocin-induced diabetic retinopathy. Let's begin by taking a look at diabetic retinopathy. It is one of the key causes of vision loss on a global scale, and it's the most common microvascular complication of diabetes. For patients with type 1 diabetes, nearly all of them will get affected by diabetic retinopathy. And for type 2 diabetes, more than 77% of patients who survive more than 20 years with the disease will get diabetic retinopathy. The main mechanism of injury is hyperglycemia-induced oxidative stress, which leads to retinal cell apoptosis. To further understand the mechanism of injury for diabetic retinopathy, we shall now take a look at the apoptotic pathways. In a cell, pro-apoptotic and anti-apoptotic proteins exist in a very delicate balance. Examples of pro-apoptotic proteins are caspase-3 and BAX. An example of an anti-apoptotic protein is BCL2. When a cell receives an apoptotic signal from within the cell itself, this will kickstart the intrinsic apoptotic pathway. An example of such a trigger is hyperglycemia-induced oxidative stress. This will lead to a drastic tilt in the balance, and there will be upregulation of pro-apoptotic proteins and downregulation of anti-apoptotic proteins. For patients with uncontrolled diabetes and persistent hyperglycemia, hyperglycemia-induced oxidative stress will be the triggering stimulus for the intrinsic apoptotic pathway. This will lead to activation of p53, followed by increased expression of pro-apoptotic proteins like BAX and caspase-3, and reduced expression of anti-apoptotic proteins such as BCL2. Eventually, this results in increased formation of apoptotic substrates and mediators. In the end, this results in retinal cell apoptosis and diabetic retinopathy. If untreated in the long term, it can even lead to blindness. The current therapies for diabetic retinopathy include vitreoretinal surgery, anti-angiogenic therapy, anti-inflammatory therapy, and laser photocoagulation. Although these current paradigms are helpful in treating diabetic retinopathy, they generally focus on the advanced stage of the disease and are highly invasive. They can potentially cause severe complications such as cataract, glaucoma, vitreous hemorrhage, retinal detachment, and even loss of vision. Now, allow me to introduce tocotrienol-rich fraction, TRF. It is a vitamin E analog derived from palm oil, and it's shown to be a very potent chain-breaking antioxidant in several studies. Therefore, it possibly possesses a therapeutic value towards oxidative stress-induced diseases, for example, diabetic retinopathy. To the best of our knowledge, no study has yet to be done on the effect of TRF on the expression of retinal pro- and anti-apoptotic proteins in animals with diabetic retinopathy. Therefore, this brought about our research objective to elucidate the effect of TRF on retinal cell apoptosis in rats with streptozotocin-induced diabetic retinopathy. Here's the methodology. Sprague Dawley rats were divided into two main groups. The first group received intraperitoneal injection of sodium citrate buffer. This group was group N, the control group treated with vehicle. The other group was given injection of streptozotocin to induce diabetes. This diabetic group was further subdivided into groups DV and DT. DV was the diabetic group treated with vehicle, and DT was the diabetic group treated with TRF. The rats went through a 12-week treatment period. Throughout this time, the rats were given daily oral gavage. Groups N and DV received the vehicle, which was olive oil. DT rats were given TRF. Every week, the body weights and blood glucose levels of the rats were monitored. After 12 weeks, the animals were sacrificed by sodium pentobarbital, and their retinas were collected. The retinas were used in the Bradford assay for measurement of total retinal protein, and ELISA was performed for the measurement of expression of caspase-3, BAX, and BCL2. Finally, the data were analyzed using GraphPad Prism 8 software. Just to recap, N is the control group, DV is the untreated group, and DT is the treated group. Now, let us go to the results and discussion. This slide shows the blood glucose levels of rats over the 12-week treatment period. For the control rats, blood glucose levels were maintained below 10 millimoles per liter throughout the 12 weeks. The diabetic rats had blood glucose levels ranging from around 25 to 35 millimoles per liter across all 12 weeks, and this was very significantly different compared to the control rats. This result is important because this shows that the effects of TRF we observed were not secondary to a decline in blood glucose levels. The protective effects of TRF against retinal cell apoptosis and diabetic retinopathy were evident despite persistent hyperglycemia. This next slide shows the effect of TRF on the expression of apoptotic markers in the retinas of rats. For rats with untreated diabetes, DV, there was higher expression of caspase-3 and BAX and lower expression of BCL2. However, when treated with TRF, the expression of caspase-3 and BAX was reduced, and the expression of BCL2 was increased. Importantly, treatment with TRF caused restoration of the expression of these apoptotic markers close to that of control rats. The BAX to BCL2 ratio was the highest in the untreated group, whereas for the control and treated groups, the ratio was similar. So, what does all of this mean? Caspase-3 and BAX are pro-apoptotic proteins, and BCL2 is anti-apoptotic. The BAX to BCL2 ratio is the main determinant of whether a cell lives or dies. Therefore, the results on this slide suggest that TRF reduces retinal cell apoptosis. To conclude using a diagram, with administration of TRF, there is reduced hyperglycemia-induced oxidative stress. Thus, there will be decreased expression of pro-apoptotic proteins and increased expression of anti-apoptotic proteins. As a result, retinal cells survive and there is preservation of visual function. In conclusion, treatment with TRF was associated with reduced expression of pro-apoptotic proteins and increased expression of anti-apoptotic proteins in rats with streptozotocin-induced diabetic retinopathy. This suggests that TRF reduces retinal cell apoptosis in rats. The protective effects of TRF against diabetes-induced retinal cell apoptosis in streptozotocin-induced diabetic rats were evident despite persistent hyperglycemia. We recommend that further studies investigate the effect of TRF on specific cell types in the retina and examine other potential mechanisms involved in the protective effect of TRF against diabetic retinopathy. Also, further studies can be carried out with a topical formulation of TRF with significant bioavailability in the posterior chamber of the eye. Here are my references. Thank you very much.
Thank you. Can I request Prof. Ong to pose a question?
Yes. Miss Goh, very neat and comprehensive presentation of your work. Very good. Well done. I would just like to clarify. You measure the apoptotic protein by ELISA using your as a measurement parameter. I just wonder, this is more like a comment rather than a question. When you sacrifice the rats, when you isolate the retina, why don't you also, look at, say, cell viability, because you are looking at the cell depth in retinal layer, isn't it? So I just wonder why you only use one measurement tool, one measurement parameter. You can actually, at the same time, also look at, say, morphology, to look at the cell apoptosis. So what do you think?
Thank you, Prof, for your comment. My project is actually a part of a larger project. So other aspects of the retinal cell apoptosis has already been done by my other seniors. So for my part, it's just on the ELISA part for the apoptosis to measure the markers.
Oh,
Yep.
okay. Okay.
Yeah.
So what sort of parameter you are also looking at your group when you look at the cell apoptosis in retina?
Yeah, so before they sacrificed the rats, they also did behavioral tests for visual function as well. And
Okay.
H&E was also done. And
Okay.
to address the other pathways involved in diabetic retinopathy, such as the neovascularization and proliferation of the vessels, they also did ELISA on the VEGF and all those. Yep.
Oh, okay. Okay. So your work only focus on,
the
level of apoptosis protein level. So it's a small puzzle of a bigger one. Okay. That's all from me. Thank you.
Thank you very much, Prof. Thank you, everyone.
Thank you, Ms. Goh You, and thank you, Prof. Ong. Let's move to the next one, which is Ms. Nurellya Faqhiraah binti Aziz from UKM.
Greetings, I am Nurellya Faqhiraah binti Aziz, a postgrad student from University Kebangsaan Malaysia, and I would like to present our study on S-allele limits cardiovascular injury induced by isoprenaline in overactomized rats. Did you know, apart from getting trouble to fall asleep, constant joint pain and hot flashes, postmenopausal women are also facing with increased risk of getting cardiovascular disease, more so those with comorbidities. Hormone replacement therapy has been shown to have unwanted side effects. But fret not, here is a promising result for all of them. An active compound found in each garlic extraction called S-allele has been studied to regulate mitochondrial survivability, hence leading to better cell survivability. It also reduces oxidative stress and cell injury, modulates the hypertension culprit, as all of us know is called as ACE81 Aziz. Lastly, it also limits cardiac hypertrophy and fibrosis. These cardioprotective mechanisms have been studied to be majorly contributed by the cystione gamma-lyase hydrogen sulfide gas, Aziz. So this gas so transmitted doesn't only facilitate with the vasodilation, but also helps with the transcription of antioxidant. So, the aim of our study is to investigate the effects of S-allele on overactomized rats with myocardial injury. To mimic the postmenopausal condition among humans with the depletion of circulating estrogen, the female rats were initially surgically overectomized, followed by 3 weeks of recovery. Then, the rats were injected with isoprenaline subcutaneous injection with the dose of 85 mg/kg twice with the 24 hours interval. After that, the rats received oral gavage treatment of either SAC with the dose of 100 mg/kg or distilled water for 7 days. Finally, the rats were sacrificed and the hearts were cannulated by the Lagendorf isolated perfuse system for cardiac function measurement. Throughout the experiment, blood pressure and electrocardiogram were monitored, and blood were withdrawn for estradiol and troponin T measurement. To evaluate the success of the overectomy surgery, uterine was weighed and serum estrogen were measured. Uterine weight is seen to be reduced in all of the groups except for sham, while as for the serum estrogen, it seemed to have a pattern of decrease among the OVX group compared to the non-OVX group. This is because uterine atrophy is because of the depletion of circulating estrogen that happens after the overectomy surgery. Before jumping to the validation of myocardial injury induction, here is ways to interpret the electrocardiogram result. The most important highlight here is the QRS, which represents the ventricular contraction, and it ends as the T recovers to the straight line, which is called as the isoelectric point. The result here shows that the RR interval and QRS interval is reduced compared to the non-MI, meanwhile it is increased compared to the non-MI and QT interval. These are the effects of isoprenaline in increasing conductivity and contractility, hence shortening between one cycle of ventricular contraction to another, and lengthening the relaxation as a result of positive lucitropic effect of isoprenaline. The increased serum troponin T in MI group right here shows that it could be increased due to its release because of the cell injury caused by isoprenaline. In oxidative stress study, only glutathione showed a significant increase in overactomized with MI group supplied with SEC, showing that SEC might have exerted its effect on increasing antioxidant level that might have been possibly through the mechanism of cystathionine gamma lyase as formerly mentioned before, an enzyme that catalyzes the production of endogenous hydrogen sulfide gas that lead to increase of antioxidant release. As we studied, only a pattern of increase is seen in CSE activity level in those hearts supplied with SEC. However, no alteration is seen in malondialdehyde levels at all. That is an oxidant product. As for cardiac function, no alteration is seen in left ventricular developed pressure and maximum contraction rate, although systolic blood pressure shows a reduction after receiving 1 week of SAC treatment in OS group right here. This is probably through the mechanism of SAC ability in modulating the ACE-AT1 axis, hence preventing the increase in blood pressure. We also did picroseries rate staining to detect the collagen deposition. Clearly, the MI-induced heart shows a prominent pink staining heart that represents the collagen deposition. This is because isoprenaline causes cell death, and this collagen deposition acts as a compensatory mechanism of the heart to maintain its elasticity and integrity. In SAC-supplied hearts, the collagen deposition are seen to be controlled, which is good because excessive collagen deposition indicates poor prognosis after myocardial injury. As to summarize my whole study, SAC from this cardiac extraction has shown to increase the antioxidant level, regulate the blood pressure, limit cell injury and fibrosis in overactomized rats with myocardial injury. So, as to conclude everything, SEC may have limited injury induced by the isoprenaline, but more molecular study is warranted to confirm its mechanism in cardioprotection. May it be because of the cystathionine gamma lyase and hydrogen sulfide? Alright, that would be all from me. Thank you. Stay home and stay safe.
Thank you. Can I invite Prof. Liz?
Thank you for that interesting talk. Can you, from looking at those graphs, it looks like you didn't see a lot of significant changes when your oxidative stress. Is that the case? And can you explain that if it is?
Yes, thank you for the question. Yes, I can't deny there are not many significant changes, but we are actually trying to study it in deeper by measuring the protein expression. It could be the case that the SAC might not be was not in a higher dose or not given in a longer time. So yeah, I think that could be the possible reason.
You also didn't see a significant effect of the estrogen levels decreasing. Was that I mean, it looked like they were going down, but it wasn't significant. Is that a validated time frame for looking at ovariectomy and estrogen levels?
According to my literature, there are many time period of recovery between all the experiments, but most of them they did more than 4 weeks until they reached that significant changes between the non-overactomized and the overactomized group.
And how long was yours?
It was just only for 3 weeks before it was given the treatment of SAC.
Right.
Yeah.
Thank you.
All right. Thank you.
Thank you, Prof. Thank you, Nurellya. Let's move on. We give a few minutes for judges to mark, and then we move to the next presenter. Just wait for a few moments. Our next presenter is Mr. Salah Al-Shehade from University of Malaya.
Hi everyone, I am Salah Shehade. I am here to talk about the potential therapeutic mechanism of action of oxyphone status using system pharmacology approach. System pharmacology uses a molecular biology and biochemistry of cellular components to understand how physiological function at the cellular and organ level arise. Recently, a novel approach of system pharmacology was proposed to identify the bioactive compound, predict the related target, and illustrate the mechanism of action. What distinguishes system pharmacology is its laser-like focus on the compound and how they are working in the body. The particular implication of such compound-centered approach are exciting a new target, a new screens, a new marker, and a new understanding of the mechanism of action. Our plant of interest is Oxyphone aristatus, which is a popular herbal in South Asia. It's frequently used for treatment many diseases, including renal inflammation and kidney stone and obesity. Several studies confirmed the therapeutic benefit of its bioactive compound, such as rosmarinic acid. In this study, we used system pharmacology approach to predict and analyze the molecular mechanism of action and to illustrate the targets and cellular pathway of the bioactive compound of Oxyphone aristatus. Oxyphone aristatus compound were identified using traditional Chinese medicine system pharmacology database beside lecture review. Then, the compounds were filtered based on the oral bioavailability. The corresponding human molecular targets were predicted using two different platforms. Finally, the pathways enrichment analysis were performed. 68 bioactive compounds of Oxyphone aristatus were filtered out of 99. A total of 876 potential targets were also predicted. First, we analyzed the potential target by reactome tools to figure out the main involving diseases. As shown in the figure, many diseases are involved significantly, including disease of programmed cell death, diseases of neuronal systems, disease of single transduction and second messenger, disorder of developmental biology, disease of immune systems, and disease of homeostasis, and also infectious diseases. Further, to get a sight of the molecular mechanism of action, we analyzed the enrichment pathways using Cake tool. We have found a significant involving in 106 pathways. The showing figure represents the top 15 results. Many of those pathways related to cancer, like prostate and colorectal cancer. Also, metabolic disorders was shown like non-alcoholic fatty liver disease, insulin resistant, type 2 diabetic, glucagon signaling pathway, and fatty acid degradation. And inflammation-related pathway was also shown, and many others. To comprehensively investigate the interaction between a herbal bioactive compound and their related biological process, we use gene ontology tool to explore the biological process that are associated. The number of genes involving in the target pathway is an indicator but not a determinant factor, as the cellular pathways can interact with each other. As you can see in this figure, most of the pathways are related to oxidative process and cell death, which is a confirmation of the previous analysis. Oxyphone aristatus could be mediated pharmacological effect of complex diseases, including metabolic disorder and cancer like prostate and colorectal cancer, through multi-target and various signaling pathways, including like kinase, protein activity, and immune system-related pathways like T and beta cell receptor, necrosis alpha, and natural killing. In addition, various compounds appear to activate the same downstream targets, therefore exhibiting the same cellular function. the system pharmacology open-end approach, which could be used to predict the molecular mechanism of action. However, further experimental investigation is required. Those are the two main references that we based on our study. Thank you all for your listening.
Thank you very much. Can we have Prof. Ong again?
Okay, yeah, Mr. Salah, thank you for your presentation. I'm not that clear from your presentation on a few things. First one is how do you when you do your initial screening, how do you started off with 99 compound? How were those 99 compounds identified?
Yeah, thank you for your question. So this is one of the initial steps. The compound were first articulated on the literature review, featuring the previous study that studied the OCON and listing out the compound and the other tool. The main tools that use is database. Recently there is many database, mostly are in Chinese database that main is to clarify and to think the compound and their active compound. And actually, those databases are well known and can be counted on.
Okay, so yeah, it sounds to me that these compounds are the known active compound present in the herbs from the database.
Yeah.
Okay, and now, yeah, okay. Second question, and now that you have identified quite a lot of pathway, actually, probably hundreds, and many of these pathway are generic. For instance, pathway associated with oxidation reduction, pathway associated with protein phosphorylation. So what's next? How then from this 100 of pathway you are able to then focus or zoom into the disease model you want to study?
Yeah, this is the first step to screening in general, how is the plan is working and what is the main diseases that could be targeted, treated by this plant. And actually it's pretty much those pathways are matched out with the previous study. For example, the obesity and cancer research. This study was done mainly to confirm if this plant could be work on non-alcoholic fatty liver disease, which our main disease of interest. So the next step was to confirm this result in vitro and in vivo, studying especially on non-alcoholic fatty liver disease. And actually, this result was pretty much matched and in line with the previous reviews.
Okay. So that means you will focus more on metabolic and cancer. Am I right? Is that your next step?
Correct.
Okay.
Correct. Especially the fatty liver disease.
Okay. That's all. Thank you.
Okay. Thanks, Alan. Thanks.
Thank you, Prof. Ong and Mr. Salah for that. Let's move to the second last speaker for this session, SITI FATIMAH binti MUKHTAR.
Assalamualaikum warahmatullahi wabarakatuh, and hi everyone. I'm Fatimah. I'm going to present on telemic probabilistic connectivity with cerebral cortex in spastic cerebral palsy. This is my presentation outline. Let me introduce you with some keywords of this research. Cerebral palsy is a neurological disorder caused by lesions in the immature brain. CP is the most common motor disability in childhood, but it is unclear whether the pathways between thalamus to motor cortices are disrupted in spastic cerebral palsy. We use diffusion MRI to look at the microstructures of white matter tracts. In vivo, no need for postmortem. It can also generate a quantitative measurement in the form of connection probability index, which refers to white matter tracts integrity. The objective of this study is to investigate the structural connectivity between thalamus and motor areas of cerebral cortex and its correlation with gross motor function measures in specific cerebral palsy patients. There are three specific objectives of this study. First is to describe the pattern of distribution between thalamus to primary motor area, supplementary motor area, and premotor area. Second is to determine the correlation between the connectivity with GMFM score. And the third is to postulate the thalamus according to its connectivity to motor cortices. Let's have a look at the methodology of this study. We use probabilistic tractography done on diffusion MRI data. Then a quantitative measurement called connection probability index was measured between thalamus to each motor cortex. Correlation was performed between the connectivity with GMFM score. Then the thalamus was further parcellated or segmented into areas that corresponded to its connection with specific motor cortex. Diffusion MRI data was processed and analyzed using a software called FIMRIP software library. For tractography, we have one seed area, which is thalamus, and 6 target regions, which are primary motor cortex, 4 areas of premotor cortex, and supplementary motor area. Let's have a look at the results of this study. The pattern of thermocourtical connectivity in cerebral palsy was variable according to the patient's clinical presentation. These are the graphs for each CP patients. The blue graph refers to the left connectivity in the left hemisphere, and the orange graph refers to the right connectivity in the right hemisphere. For CP1 subject, the left connectivity was lower than the right for all targets. This corresponded to the diagnosis of right hemiparesis. Let's have a look at the results for second objective. There was no significant correlation between thermocourtical connectivity and gross motor function measures. For the third objective, the result was in the form of images of segmented thalamus. For healthy control, the cluster that has positive connection to primary motor cortex, which is the green area, is related to the lateral group of thalamic nuclei. For postulation of thalamus in cerebral palsy, a striking feature was that there was a presence of cluster that has positive connection to supplementary motor area, which is the red area here. That was none in healthy control. Moving on to the discussion part. Large variabilities of connection probability index within and between the cerebral palsy patients, because each lesion brain was unique according to their clinical presentation, but some findings were not consistent with the clinical characteristics because several neuronal processes has occurred post-brain injury that leads to a functional reorganization of the neurons in the brain. Hemiplegic patients in this study showed a connectivity pattern that corresponded with their clinical presentation. They have lower values of connection probability indices in one hemisphere that caused contralateral motor impairment. This could be related with the perinatal stroke that is common in unilateral CP. For the second objective, there was no correlation between the connectivity with GMFM score because it is related with the large variabilities in the connection probability index in CP patients due to the morphological changes as the compensatory mechanism in the brain injury. Our small sample size could also affect correlation analysis, and other factors could also influence the GMFM score, for example, other tracts like corticospinal tracts or exercise therapy. For postulation of thalamus in healthy control, cluster that has positive connection to primary motor cortex were associated with a lateral group nuclei because it contained main motor relay station, which are ventral anterior and ventral lateral nuclei. For postulation of thalamus in cerebral palsy, presence of cluster that has positive connection to supplementary motor area because it gives a great contribution for motor function as the compensatory mechanism in brain injury. Here comes the concept of neuroplasticity, whereby the brain undergo morphological and functional modifications post-injury. In conclusion, there were vast variabilities and unique neuronal connections in cerebral palsy corresponding to the patient's clinical presentation. There was also an evidence of neuroplasticity as a compensatory mechanism for the motor deficit in CP. These are the list of some of my references. With that, I thank you.
Thank you, Siti Fatima. Can I invite Prof. Elizabeth Davis for a question?
Thank you very much. Can you tell me a little bit about the causes of cerebral palsy? Is it usually related to some sort of damage at birth, or is there a genetic component, or do we know?
All right, thank you very much for your question. So there are risk factors for CP. The main cause is caused to the immature brain. Like the cause is due to the lesions in the developing brain. So usually it was happened before birth. And there are some risk factors like prematurity and the HIE, hypoxic ischemic encephalopathy, or the perinatal events that may cause injury to the brain. So usually the cause is before the brain fully developed, like before the age of 2 years old, usually the cause of CP.
And where do you see your research going next? What's the next thing you're going to be looking at?
For this current topic, we can, I think I would like to do some intervention for the patients. So like the therapeutic intervention or the therapy. So we can measure the motor function before and post intervention to look at the changes of the white matter tracts pre and post intervention. So yeah, we can have a look at the effects of intervention that may improve the motor outcomes of the patients.
Thank you.
Thank you.
Thank you, Siti, and thank you, Prof. Elizabeth. Let's move to the last presenter of the day, not for the day, for the session, sorry, which is Hann Yih.
Very good day to everyone. I'm Hann Yih, third-year medical student from IMU, and the topic of my presentation today is the effect of resveratrol on glutamate clearance and visual behavior due to excitotoxic retina injury in rats. Now, let's look at the relationship between glutamate and glaucoma. So glutamate is the major excitatory neurotransmitter in retina, but excessive glutamate neurotransmission will lead to excitotoxic injury. So normally, when glutamate released from presynaptic cells will bind to its receptor on the postsynaptic cells, which leads to the increase of calcium ion concentration in the cell. The increase of calcium ions concentration will lead to action potential. The glutamate will then clear it out by two routes. The first one is direct uptake from the presynaptic cells, and the second route is the uptake by glial cells through two transporter, which is EAAT1 and EAAT2. But if there is excessive glutamate, this will lead to excitotoxicity. So when there is excessive glutamate, it will lead to excessive calcium ions concentration build up, and these calcium ions will then accumulate in the mitochondria, which will lead to the leakage of cytochrome C. This cytochrome C will bind with APAF1, forming apoptosome, and apoptosome will then activate caspase 9, which then activates caspase 3 and activates apoptosis, and this will lead to glaucoma. And that's why in our research this time, we like to explore more on this substance here, which is called resveratrol. It is widely known for its neuroprotective properties, and it was shown to counteract the hyperglycemia-induced reduction in expressions of EAAT1 and EAAT2 and glutamate clearance in the research in 2016. And it still remains unknown if resveratrol affects EAAT1 and EAAT2 expression and protects against excitotoxicity under non-diabetic condition. Now let's look at the objectives of the project. The first one is to determine the effect of resveratrol on glutamate clearance by measuring the retina glutamate level and EAAT1 and EAAT2 expression using ELISA in rats. And the second one is to determine the effect of tranresveratrol on glutamate-induced changes in visual behavior of rats using open field test and chamber mirror test. And the third one is to determine the effect of tranresveratrol on glutamate-induced changes in retina morphology in rats using H&E staining. Now, this is the study design. So we'll separate the rats into 5 different groups, which is the untreated, the intravitreal vehicle, the intravitreal glutamate group, and intravitreal tranresveratrol, followed by glutamate 24 hours later. And this is our pre-treatment group. And also intravitreal glutamate, followed by tranresveratrol 24 hours later, which is our post-treatment group. And on day 6, the rats went through open field test. On day 7, they went through chamber mirror test. And on day 8, I sacrificed them and their eyes were enucleated. And I used ELISA to check for retina expressions of glutamate, EAAT1 and EAAT2 expressions. And I used H&E staining for retina morphology. And I used GraphPad for statistical analysis. We have settings for open field test and the settings for chamber mirror test here. Now let's look at the result and discussion part. So we can clearly see that rats in intravitreal glutamate injection, expressions of glutamate transporter remains comparable to the PBS group, which perhaps is inadequate to clear up the increased glutamate injected exogenously. Hence, retinal glutamate concentration in this group is high compared to the PBS group. In TR-treated groups, pre-treatment group showed greater expressions of both transporter, and accordingly, the retinal glutamate level was significantly lower in this group. In the tranresveratrol post-treatment group, we observe a trend towards high expressions of both transporter. However, the difference from GA group was not significant. Accordingly, a significant reduction in glutamate level was not observed, as was the case for pre-treatment group. For the retina morphology, I've selected one picture to represent each group, and the different colors arrows represent different layers of the retina. Now, if you look at the data closely, we'll notice that for the GA rats, they're having the least thickness for all the retina layers, whereas if you look at the treatment group, we'll see that the GATR and the TRGA rats is having the similar results when compared to the UT and the PBS groups. And now let's look at some data from the open field test. And now this is the track plots of the test. We can see that the GA rats spend more time exploring the peripheral zone and less time in the central zone, whereas the others are willingly to explore the central zone more. Overall, there were no difference between the untreated and PBS treated group. However, the GA treated group showed high locomotor but lower exploratory behavior compared to the PBS group. The emotional status was also higher in the GA rats, but overall, this indicates elevated anxiety in GA-treated rats. In tranresveratrol post-treated rats, we observed the overall behavior closer to the PBS group, but this was more evident in the pre-treatment group that showed overall behavior very close to the PBS group. These observations are in accordance with previously published paper, one of which is listed below. These observations correlate with the difference in the expressions of retinal glutamate transporter and glutamate concentration among various groups. Now let's look at data from the chamber mirror test. And we can observe the pattern. All other rats except the GA rats spend more time in the inverse mirror chamber. According to the graph, the GATR and the TRGA rats had a similar visual status as untreated and control rats. These rats spend more time in the inverse mirror chamber. And this can be explained with a paper published by File and Height in 1978. The paper claimed that when two healthy rats are placed in an unfamiliar test environment, the overall level of social interaction is suppressed. In this case, healthy rats are able to see their reflection clearly in the mirror chamber without knowing that it is their reflection. Therefore, they escape from the mirror chamber to avoid any social interaction with the reflection. We also noticed that no zone preference were observed among the GA rats, and time spent in both mirror and inverse mirror zones were almost equal. All the rats were not subjected to prior training, and the single sessions of tests were performed over 10 minutes. Therefore, it seems that the GA rats have some degree of visual loss that affects their ability to recognize themselves in the mirror, and this is likely to result into their inability to distinguish between mirror and inverse mirror. In conclusion, treatment with tranresveratrol increases expressions of EAAT1 and EAAT2 and also reduces retinal glutamate concentration in rats. And this suggests that tranresveratrol enhances glutamate clearance and reduces excitotoxicity. We also can conclude that treatment with tranresveratrol preserves glutamate-induced changes in visual behavior in rats. And lastly, treatment with tranresveratrol preserves glutamate-induced changes in retina morphological features. Overall, pre-treatment group showed greater efficacy when compared to the post-treatment group. And this is the end of my presentation. Thank you so much for your time.
Thank you. Can I invite Prof. Ong for the question?
Yes, yeah, Mr. T. Yeah. Okay. Yeah. Congratulations on a job well done. I think I can tell there's a lot of work being done for a 7-minute video. So well done. Okay, I think your presentation is neat, concise, but there is one thing that I'm not quite clear. In your chamber mirror test, when you look at time span in a mirror and inverse mirror section for the different group, I don't really see the difference actually. When you look at the slide showing the tracing of the rats, can you sort of explain more on that?
Right. So
Yeah,
so if you look,
you say that there is differences.
yeah, so if you look back to the graph, the figures, you'll see that actually most other than the J rats, all other rats spend more time in the inverse mirror. Yeah, but whereas in the J rats, they spend equally time in both chambers.
Yeah, in the bar chart,
yes,
I think you show the difference, but I think the earlier slide where you show the tracing, I seems not to get that picture. You say glutamate group, is that right? Glutamate treated group spend about the same time, but
no, it's I think it's because
I can't see that clearly
right. I think it's because that's
earlier slide. Yeah.
Yeah, I think it's because that's a track plot, which means it's track where the rats is. But then that track plot doesn't indicate like how much time they spend in the chamber. So I guess the bar chart will explain more, can explain better with the time spent in each chambers.
Okay. Okay. And also, I guess that the figure in your bar chart is a sum of a few observations, isn't it? So I think that will show the differences clearer. Huh?
Okay. Yeah.
Okay. And also, I noticed your administration of resveratrol is through. Is it through oral administration?
No, it's through intravitreal.
I can't recall.
It's called. It's through intravitreal. Yes. Yeah.
Okay. So in that case, you are looking at the local effect, local application. Is that right?
Yes.
So okay. So I'm just not clear about that. Okay. And another one is it seems like you don't have a positive control in your study. A known agent that can be used as a comparison to reduce, say, retinal damage?
Yeah, no, there's none.
No, probably it would be good if you have one. Probably that's for coming work.
Yeah.
So it will be nice to have one. Okay, that's a comment. Okay, I think that's about all from me. Well done. Well done.
Okay. Thank you very much, Prof. Thank you.
All right. Thank you, Prof. Thank you, T. So that's basically end the session. We'll allow judges for a couple of minutes to complete their scoring. And I urge the presenters to stay here. We're just gonna take a quick group photo. So I request everyone to on their video here. So we take a group photo with judges. Okay, Miss Go, if you can also on your video, and also Ilya. Miss Nurellya and Miss Go. Are you around? If not, we'll just take the photo with this. I'll do the sprint screen. 1, 2, 3. Someone is coming up. Miss Ilya, are you coming? All right. 1, 2, 3. One more. 1, 2, 3. Okay, thank you. Okay, we'll end the session, and those who are presenting in the next session, we request them to be before time, so we can move you to the panelists. Thank you again, our judges, especially Prof. Elizabeth Davis and Prof. Ong, for your kind contribution to this conference. We'll see you guys in the conference. Thank you. Bye, everyone. We'll see you in the next session.